simulation that allows one to study dynamics on timescales beyond that which one can normally study within classical molecular dynamics, called accelerated MD. I'd also like to thank the previous speaker, because in his last 3 or 4 slides, he actually gave me an enormously good introduction to what I'm going to talk about. I could have asked for a better introduction, to be perfectly honest. So I know you're all tired, so I'm going to try and be gentle. But we all know that many biologically important processes, such as enzyme kinetics and ligand binding, occur on microsecond to millisecond timescale. That includes kinases, of course, as we just heard. And the study of such slow timescale dynamics remains a challenge to experimentalists and theoreticians alike. Now, certainly through a variety of X-ray crystal structures and diversity of structures that one observes, one can get models out about potential long timescale dynamics. And also through NMR, which I'm going to talk about a bit as well, you can also extract information about long timescale dynamics. But what we would really like is to have a fully atomistic representation of a mechanism with a free energy barrier, estimation of rates and the like. And ideally, you would say simulation would be very good at doing this. The problem we have with simulation is, despite the fact that computational power has increased and our codes are much more efficient, classical MD is limited still to timescales of tens to hundreds of nanoseconds, with a few rare exceptions. For example, David Shaw's group, as I'm sure you'll hear on Sunday, who can go up to maybe a microsecond or so. So in light of this, we have been developing over the last several decades novel methodologies that allow us to somehow accelerate the amount of conformational space that we can sample within classical MD simulations. And the web method that I'm going to present is a method called accelerated MD. So my outline is going to be that I'm just going to present the basic idea of what we're doing in accelerated molecular dynamics, and then I'm going to try and prove to you that it actually works by showing you a very simple system, ubiquitin, everybody's favorite molecule after kinase. And after that, having shown you that it works, I'm going to give you an example of how we can study slow, that is micro to millisecond functional dynamics in proteins, and also how we can look at natively unstructured proteins. So in accelerated molecular dynamics, we define a boost energy or reference energy that lies above the potential energy surface of the system. And then during the simulation, at each step, we calculate the potential energy where we are in the configurational space. If that potential energy happens to be above the boost energy, then we leave the potential energy alone. But if the potential energy is below the boost energy, we add a continuous non-negative bias potential. And the amount of energy that we add to the potential energy surface depends on this energy difference. So therefore, if you're in a low energy region of the potential energy surface, we're going to add a large amount of energy to the system. Whereas if you're in a high energy region of the potential energy surface, we're going to add a low amount, we're going to add a small bias to the system. And we can control how much we raise and flatten the potential energy surface by an extra parameter here called the acceleration parameter. So to make this a little bit clearer, the picture says more than 1,000 words. So this is the black line represents the hypothetical one-dimensional potential energy surface. And our boost energy I've set at 12 kilocalories per mole. Now in an ordinary simulation, a classical MD simulation, we'd be sitting here at the bottom of this potential energy well, and we'd basically be stuck there. We'd be moving backwards and forward, and we'd have to do a simulation for a very long period of time before we could somehow jump over this relatively large energy barrier of some 9 kilocalories per mole to get into different conformational microstates. But as we accelerate by using this alpha parameter to raise and flatten the potential energy surface, we keep the underlying shape of the potential energy surface, but you can see that we're significantly reducing the energy barriers between the different low energy microstates. And therefore, if our simulation were to be performed on a modified accelerated potential, such as, for example, the blue line here, we would see that we could traverse these energy barriers much quicker and therefore enhance the conformational sampling. And that's the basis of accelerated molecular dynamics. Now, the advantage of AMD is the fact that because we know at each point how much we're raising and flattening the potential energy surface, is we can easily reweight our molecular conformation in order to get back to the correct canonical Boltzmann distribution. In other words, AMD is a robust free energy sampling approach. The disadvantage of AMD is the fact that whilst we are efficiently sampling conformational space, it is difficult to extract a meaningful estimate of the exact timescales of the motions that we're observing. Now, in order to justify that AMD works, there's been a lot of work in Andrew McCammon's group several years ago on small polypeptides, which has shown that you indeed can get small polypeptides to change between different conformational states. But simulating a polypeptide is one thing, simulating a protein is something different. So we need to have some experimental data which is going to allow me to see how well my AMD simulations work at probing long timescale dynamics. And so I go back to nuclear magnetic resonance spectroscopy. And there are various experiments that have been developed, as the previous speaker outlined in one of his slides, and they probe timescales in proteins on different timescales. I'm going to be looking at two types of experiments, dipole-dipole auto-relaxation and so-called residual dipolar couplings. There are two different experiments. They probe protein dynamics on different timescales, but they're both based on the same physical phenomenon, and that is the so-called dipolar coupling. So the basic idea is that in any NMR experiment, you will induce magnetisation on a nucleus. Usually we induce magnetisation on the nitrogen atom, the amino nitrogen in each residue. At that point, you've brought the system away from equilibration. Somehow the magnetisation is going to have to be dispersed across the entire molecule. And in order to do that, the magnetisation is transferred through a dipolar coupling mechanism, which can be described by a second-rank tensor. And the magnitude of this dipolar coupling is dependent on a whole series of parameters. Some of them are chemical parameters, such as the jar magnetic ratio of the two nuclei that are involved. Then you have physical parameters, such as the interatomic distance between the two nuclei. It also depends on the strength of the external magnetic field vector, and also on this angle subtended between the NH bond vector and the external magnetic field. Now, in the case of 15N dipolar coupling, it turns out because the proton has a jar magnetic ratio which is 10 times larger than any other spin-active nuclei, that the predominant relaxation mechanism is indeed the relaxation between the amino nitrogen and its proton. And the relaxation that we're going to measure, the transfer of magnetisation, is ostensibly due to, is mediated by dynamic motions of this NH bond vector. And this is what we're measuring in dipole-dipole auto-relaxation. We're measuring ostensibly over time how the magnetisation on that nucleus, on that nitrogen nucleus, decays. And that phenomenon, known as relaxation, is mediated by both the tumbling of the molecule, but also by the internal dynamics, how much this reorientational motion of the NH bond vector, and therefore of that particular peptide residue, is occurring. And we can describe this and also calculate it in simulations using a so-called correlation function. So this correlation function, as I said, is expressed in two terms. There's a rotation diffusion term and an internal dynamics term. The rotation diffusion term we consider to be an exponential decay function, and we can see that basically for a small globular protein such as ubiquitin, by the time you get out to 10 nanoseconds or so, the system has completely relaxed, and that's just through tumbling. When we look at the internal correlation function, you see that you get a different correlation function. You start at 1, it decays to a sort of plateau. Now this plateau is called the order parameter, and it's a quantitative measure of how much dynamics is occurring. But of course it's limited to timescales of the rotation diffusion. You can't probe internal dynamics beyond about several nanoseconds in this case, because the tumbling of the molecule has basically already relaxed the system. So spin relaxation is useful for probing dynamics on timescales of picos and nanoseconds. Now, in an isotropic solution, you can't measure the actual dipolar couplings, but it turns out that due to this tumbling effect, they average to 0. But it was pointed out by Jim Prestegard about 15 years ago, 10 years ago now, that if you could put a molecule in a weak alignment media, you could get a very small percentage of the molecules to partially align within that alignment media. It could be a liquid crystal or a phage. And just from that 1% or so of molecules that align, you could measure a dipolar coupling, which now becomes called a residual dipolar coupling. And these residual dipolar couplings, physics tells us, are providing information about both structure and dynamics, but are sensitive to dynamics up to timescales of 1 over the magnitude of the coupling, which basically for an NH residual dipolar coupling would mean about 10 milliseconds. So we have two experiments here. We have spin relaxation, which is probing picosecond to nanosecond dynamics, and we have residual dipolar couplings that are probing dynamics up to 10 milliseconds. So the basic idea of the project for ubiquitin was to perform a series of accelerated molecular dynamic simulations and systematically increase the conformational space that we're sampling by accelerating more and more aggressively. And then for each of our molecular ensembles, we back calculate the RDCs using a method called single value decomposition to identify the optimal alignment tensor. And then we can perform a statistical test called the Q-factor test to determine how well the theoretical and the experimental RDCs agree. And we can compare our accelerated molecular dynamics results to standard classical ND simulations. The idea behind this being that at some point we should sample enough conformational space that we're actually getting out to millisecond timescale dynamics. And at that point we should be getting the best residual dipolar coupling data. So this is what we see in ubiquitin. This is the order parameter that I talked about as a function of the acceleration rate. The top line is basically a standard MD simulation, and all the other lines, as they're significantly decreasing in certain regions, these are increasing the acceleration rate. So because the order parameter is decreasing, we're saying that in certain regions we're always seeing substantially more dynamics. And you can see that it's a very heterogeneous distribution. There are two particular regions where we're seeing a large amount of dynamics. The first loop region and around residues 46, 47. Now the problem we have is, as I said, we don't know the timescales that we're probing. So we can carry on accelerating as much as we want to, but at the end of the day we don't know how much conformational space is really appropriate for millisecond timescale dynamics. So what we do is we then calculate our residual dipolar couplings. We have a large amount of data available to us. We have RDCs measured in 23 different alignment media from Christian Griesinger, and we calculate the Q values. So basically, the smaller the Q value, the better the theoretical result. And we start off at a boost energy of 0, which is a standard empty simulation, with a Q value of 0.21, and you can see that the Q value improves. It gets smaller, so we're getting better results to our theoretical RDCs. And then at some point, at some level of acceleration, it gets worse again. So it appears that there's an optimal conformational space sampling ensemble that we've acquired at this particular acceleration. Now, it turns out that we can look at this is just an idea of the sort of agreement that we're getting when we compare theory with experiment for RDCs in different alignment media. But it turns out that, of course, we can calculate other experimental parameters as well. One of them are scalar j-couplings, another NMR interaction which is sensitive to dynamics on millisecond timescales. And again, we do the same sort of SVD approach. In this case, we're trying to optimize the corpus parameters to look at the backbone 3J scalar couplings. And again, our Q factor, we see, improves. And again, it's around this boost energy, E boost minus V torsion of 250 kilocalories per mole, that we seem to be getting like an optimal agreement, which was exactly the same for RDCs. Now, in order to rigorously prove this, we started doing a very long standard classical ND simulation of ubiquitin. And by the time we got out to 120 nanoseconds, when we did our RDC Q factor analysis, we ended up with a value of 0.175. And if we went back, oops, if we see at our lowest acceleration rate, we actually get a Q value of 0.17. So we were saying, okay, anyway, it looks like at the lowest acceleration level, we're actually seeing the equivalent of 120 nanoseconds of standard classical molecular dynamics. And if this is true, then let's see what the order parameters look like. See if they're the same. So here you see the AMD result and the 120 nanosecond MD result, and they're essentially identical. So what we've shown is that whilst the AMD simulation was only done over effectively 8 nanoseconds, we've actually managed to reproduce a conformational ensemble, which was the equivalent of 120 nanoseconds of standard classical MD. And as we increase this acceleration level further, we're going to get an enhanced amount of sampling, and it is non-linear. It's almost exponential, essentially. And so, of course, on the basis of that, we can then take our optimal conformational ensemble, which we know is probing micro to millisecond dynamics, and we can compare the order parameters to our standard 15N relaxation. And you see that, again, there's certain regions where we're seeing order parameters that are much lower. So this basically is a means that we have of very efficiently obtaining information about timescales of dynamics on micro to millisecond timescales. And in order to fill in for the NMR experts here, we see a global improvement in all the RDCs in all alignment media. And it's not just the RDCs, the residues that are displaying slow timescale dynamics are showing improvement. We see an improvement in all the residues. And this is important because what's happening is that as we sample more conformational space, the time average shape of the molecule is changing. And that means that our estimation of the alignment tensor is improving. And because our alignment tensor is improving, that means that, our residues, even those residues that do not show long-term scale dynamics, they also show an improvement in the residual dipolar coupling. The j-coupling improvement, as you probably noticed, was quite a bit smaller. This is because in the j-coupling, it's really a local effect. So it's only the residues that are displaying slow timescale dynamics that show an improvement. Our optimal AMD ensemble does not violate NOE data, which is very important for NMR people. And on approaching the optimal conformational sampling space, the average structure that we get approaches a single copy representation obtained from ADBAX, this so-called 1D3Z structure, which is using all the available NMR data as restraints. So it seems that even though we're sampling more conformational space, we're actually sampling around an average structure that matches up very well with probably the highest resolution NMR structure that actually exists. And the other interesting result that we got out of this is that if we started doing the standard ND simulations using the X-ray crystal structure, and we just calculated these fast timescale dynamics, these 15N order parameters, the agreement was relatively good. But if we actually start calculating order parameters in different conformational microstates that we have identified through our AMD, and then we average the order parameters using our free energy weighting, we get an improvement in the fast timescale dynamic order parameters as well. So all this data is basically telling us that our AMD approach is efficiently getting out to long timescale dynamics, and we can reproduce experimental data to a high degree of accuracy. Well, that's great, but ubiquitin is a small globular protein. I want to look at something a little bit more interesting, like functional dynamics. So I chose to look at the hemophore HasA, which is found in gram-negative bacteria. Its function, like all hemophores, is to scavenge heme and transport it to its particular membrane receptor, HasA. And we're interested in this because as ion assimilation plays a key role in microbial infection, HasA represents a potential drug target. Unfortunately, we happen to have both apo and hollow forms. And if you look at them, you can actually see that there's a huge structural transition that occurs on binding to heme. This loop region, particularly at the top here, which is open, actually folds all the way down and closes onto the heme. And then there's another loop region below that undergoes a smaller transition. But basically, this is a transition through some 30 angstroms. Well, now, we can do standard ND simulations on the APO structure, and we can see that we get relatively good agreements in terms of the fast timescale dynamics. These are the order parameters. It's interesting to note that the experimental values here are very large. They're almost 1 in the regions of the 2 loops, here and here. And this is indicative of exchange relaxation, which is affecting our spin relaxation analysis and creating an artificially large order parameter. It's indicative of the fact that there must be some slow timescale dynamics occurring in these regions. But what we don't see is any functional dynamics. When we do accelerated molecular dynamics, we insert the heme, we actually see within a relatively short period of time the entire structural transition. This is work in progress, but basically already we have enough information to basically come up with a mechanism. As we insert the heme, the first thing that happens is this lower loop lifts upwards. And by lifting upwards, it effectively causes a small structural change in the entire corpus of the protein. Importantly, this involves rearrangements of hydrogen bondings in this beta sheet at the back. And the outermost beta strand breaks off and becomes exceedingly flexible. And it starts to form contacts with the heme. And that allows basically this loop to fold over the protein corpus through a 30-degree transition, and eventually at some point we form a 310 alpha helix, and that allows this loop region to clamp down on top of the heme. So without using any restraints or the like, we actually get to an RMSD to the known hollow structure of about 1.5 angstroms. So that was an example of functional dynamics. Now I'm going to talk about natively unstructured proteins and show how AMD can be used as a robust 3D sampling approach. Now, we don't usually talk about natively unstructured proteins, mainly because X-ray crystallography isn't any good at looking at them. But if you actually start going through what these things do, they're actually quite important. They're involved in transcriptional regulation, translation, cellular signal transduction, molecular recognition, neurodegenerative pathology, and I start running out of colours. But we have a real problem with how we're going to deal with the structure-function paradigm when we don't have a structure. So the way we started looking at this was saying, well, obviously, a single structure is no longer appropriate, so we need to have an ensemble of interchanging structures to describe the conformational behaviour. And the way that we're going to overcome this paradigm is that we're going to identify rules of conformational sampling. So again, we have to find some experimental value that we can go back to in order to justify our simulations. And it turns out that these residual dipolar couplings are very useful because you can measure residual dipolar couplings in natively unstructured proteins, and they contain an enormous amount of information about the conformational behaviour. So you can see in this example from PX domain from Zendai virus, this is a partially natively unstructured protein which has a folded region and an unfolded region. Obviously the RDCs are very large in the folded region, but you can see that we can measure finite RDCs in the unfolded region. And what we notice is that first of all, we see that the RDCs have a bow-type profile. This is typical of all unstructured proteins. In this case, because it's a partially unstructured protein, you're seeing half of the bow here. Basically, in the middle of the unstructured region, you tend to see that the RDCs are larger than at the end. And we understand this by saying that in these natively unstructured proteins, in the middle of the protein, the system tends to be more elongated than at the ends. At the ends, it's waving around an awful lot, and the averaging is driving the RDC values to zero. But in the middle of an unstructured protein, due to global steric interactions, they tend to be more extended, and therefore slightly more structured, and this is therefore giving us a larger residual dipolar coupling. But you can also see, as you look along the residue sequence, that there are large variations. There's a high degree of fine structure here in these RDCs, and we consider that this is to be due to conformational propensities. So certain regions of the protein want to exist maybe as an extended beta sheet. Other regions want to exist predominantly as a polyprotein 2. Other regions maybe as an alpha helical term. And then somehow we're getting information here about this. So our aim was to try and basically simulate this. We almost gave up, essentially, initially, on the idea of ever being able to do a standard molecular dynamic simulation of this. Even AMD could not sample the amount of conformational space that these natively unstructured proteins traverse. So we went to a Monte-Carlo modelling approach initially. We went to a database of known X-ray determined PDB structures. We threw out the secondary structure elements, the alpha helices, the beta sheets. We just looked at the loop regions. And for each individual residue, we created an effective Ramahandran plot, a phi-psi database. And then using that phi-psi database, we rapidly constructed a conformer. So if this conformer had a sequence which was lysine, arginine, aspartic acid, and so on, basically we start off here in our molecular modeling protocol. We randomly take 1 point out of this lysine Ramahandran plot, and we construct the backbone geometry of that residue. We then go on to the next residue, we say it's an arginine, we randomly extract 1 point from that arginine, and we can construct rapidly the entire monomer. We also include an exclusion zone, a simple sphere, to try and prevent steric clashes. But we needed a method that was fast and quick, because once we generated the structure of one monomer, we can then calculate the alignment tensor using a code called PALAS and calculate the RDC. But we need to do this hundreds and hundreds of thousands of times over in order to get some idea of the enormous conformational ensemble that is being sampled. And then we average the RDCs. This was our initial idea, and we tried it on the Zendai virus. And remarkably enough, for such a simple model, it worked incredibly well. You can see that this is our molecular ensemble. You can see how much conformational space that we're sampling. This is why we didn't think you could do it with molecular dynamic simulation methods. But you can see that we're actually reproducing the RDCs very well in the unfolded region and also in the folded region. And that's important. The fact that we can reproduce the RDCs in the folded region means that basically the ensemble is reproducing the average shape of the molecule, and so our alignment tensor is correct. Now, when I first saw this, I was rather surprised, because even though I come from a physics background, I know that sequence in biology is very important. And the fact that we're using a database of X-ray crystal structures means we have no sequence specificity whatsoever. So in one of our PDB structures that we're looking at in a loop region, if we're interested in a serine residue, the sequence might be tryptophan, tryptophan, serine, adenine, adenine. But in another one, it might be alanine, phenylalanine, serine, serine, serine. We aren't differentiating between these. And therefore, the fact that we can reproduce these RDCs so well, which means that we reproduce not only the global shape of the unfolded part of the protein, but also the local structural propensities, seemed rather surprising. But nevertheless, we said, well, okay, let's go and look at something that everybody's interested in. So we went to tau protein, which, as we all know, is the principal vector associated with Alzheimer's. It's found in high concentrations of neurofibrillary tangles, and the mechanism for the transition from the normal to the pathogenic state remains unknown. And we did exactly the same thing as I've shown you on this endovirus. And suddenly we see that there's a bit of a problem. You can see in the experimental values that sometimes the model is actually doing very well. But there are certainly 4 or 5 regions here where the experimental value is clearly showing that there is a very significant dip in our profile, and the theory isn't reproducing this at all. And when we start looking at the sequences, well, it's a 4-repeat domain, we actually see that in these dips, we're actually seeing very similar sequences. So this one is DLKNB, DLSNB, DLSKB, and the last one is a little bit different, it's DFKDR. But we also see, when we look more closely, that there are other regions where we're not really doing that well, such as here and here and here. So, how do we overcome this problem? We can go back to a database and we can look specifically for that sequence in loop regions. Problem is, you'd like to find only about one protein that we've actually got a structure for that shows this. So we thought, well, okay, we've got this method called accelerated molecular dynamics. So we can use this to extract free energy weighted phi-psi propensities across our sequence. So we take small fragments, 15 residue fragments, we do AMD simulations, as we sample all the way down the chain, and having done our reweighting, we then calculate effective gamma handling plots for each individual residue. But this time, of course, they're sequence specific. And then we do exactly the same molecular modeling protocol, but we replace the database with our AMD result. And to cut a long story short, this is what you see. You see that we actually nicely reproduce all the little dips and everything. And the reason is because the phi-psi propensities show that these regions here actually like to form beta-1 type terms. This has been identified as well in infrared spectroscopy. And we also see that the regions here are actually getting better as well. And this is because the AMD is telling us that these regions like to form extended beta sheets. Now, when we first saw this, we thought this was a great result. We thought we'd understood the reason behind why tau would fibrillate. We thought that somehow, if you could get rid of these beta-1 terms, then these extended beta sheets would sort of all pack together, and that would cause fibrillation, and that was what was driving Alzheimer's. Unfortunately, shortly after we published this, a paper came out in PNAS, where they actually showed structural evidence that these terms are actually present in the pathological state as well. So we're not sure exactly what the reason is for these terms being there, but we assume that it's an early evolutionary attempt to prevent fibrillation. And just to show you that it doesn't just work on tau, we then looked at P53, which you all know is the so-called guardian of the genome. It's important to us because in 50% of cancer-related tumours it's found to be inactive, and it contains an N-terminal transactivation domain, which is a 93 residue intrinsically unfolded domain. And the studies that we've done on Zendivirus and also on tau showed us that the model actually works pretty well, the standard database model, for most of the protein. It's certain regions where the model has difficulties, and in general, the AMD results in the model, when we get good agreement with the experimental data, they tend to agree reasonably well as well. So the model, of course, is much, much quicker than the AMD result. So in order to speed things up, we do the model first, And when we identify regions where we're having problems, and then we use AMD to sort these things out, or at least try to. So this is what we see for the disordered N-terminal transaction domain, 93 residue protein. This is our model. You see it works really well. I mean, residues from 25 odd to, or 30 odd to 60, we actually get a really good reproduction of the data. But there seems to be a problem here around residues 22 to 25. And this is where stimulation is nice, because you do your AMD, you stick it in, and suddenly it gets better. And we have a bit of a problem in the proline-rich region down here. So we look at that with AMD, and it gets better again. And now we've actually reproduced the RDCs remarkably well using this molecular modelling AMD type approach. This 310 alpha helix that we're seeing is quite important, because this is actually where we know that MDM2 is binding, and MDM2 is the protein that regulates p53 activity. It turns out that the proline-rich region here, that this is a very rigid region. That's why we're getting very large RDCs. That turns out to be interesting as well. And we see this when we go to the full tetrameric complex. Turns out that when we look at the full tetrameric complex, we can use the same 5-side propensities that we just looked at for TAD, and we can reproduce the RDCs in this enormous complex here. So obviously the 5-side propensity weightings are the same. And now we get to understand why this region here is actually so rigid. The reason is that because in this N-terminal region, the C-terminal domain is bound to the corpus of the protein, and it's quite rigid. So if I am the corpus of p53, this bit here in this sort of bubbly shape thing, well, then this C-terminal region of this polyproline-rich region, this is essentially rigid. So this is like my arm pointing out as a rigid sort of robotic arm that's moving around like this. And at the end of my fist, then you have the flexible part, which contains the binding site for MDM2. And if you think about it, this is probably the most efficient way that p53 could sample a huge amount of conformational space without interacting with it, or without its own corpus getting in the way. So my conclusion is very simple. It's actually what's going to be the second title of my talk, which is normal timescale MD simulations for everybody. You don't have to have, Blue Gene and these sort of programs, this sort of computer power available to you. On pretty much every project that I've shown you today, I was using less than 8 processors. And yet I'm still getting out to micro to millisecond timescale dynamics. And finally, I'd like to thank various people, my present boss, Andrew McCallum, Martin Blackledge and Guillaume Bavigny, work at the Structural Biology Institute in Grenoble. These are the people that I did a lot of AMD with. Michael Nilgis, Professor Milo Delpierre and Nicola Wolff. This is the HasA project, the Pasteur Institute. Marcus Spetzlecker and Christian Wiesinger provided all the data for the ubiquitin project, and also the data for the tau protein. And Mark Wells and Professor Alan Furch at Cambridge University for providing the NMR data for p53. And I'd like to thank you for your attention.
So we have a few questions here.
I'm not so sure if I understood exactly that you modify the potential function, but then do you re-debias that later?
Yes, we...
That's what I misunderstood how you did that.
So, in order to enhance the sampling, we add on this bias potential. And now, effectively, during our simulation, the system is not moving on the original potential, it's moving along the modified potential. But at each point, we know how much we're raising and flattening the potential energy surface. And because we know that, we can take each point in our configuration space, each structure that we extract along the trajectory, and we can weight it by, it's the Boltzmann factor of the bias potential. So exp to the power 1 over RT times the bias potential. And that essentially accounts for the amount by which we're raising the potential energy surface, and therefore we're reweighting the configurational space to get back to the original canonical Boltzmann distribution.
Okay, that's clear, but how large are these perturbations? That's surprising.
That depends very much on how much you accelerate, of course. If you accelerate a lot, but it's all relative, of course, because ultimately, in order to achieve any decent acceleration, your boost energy is essentially always going to be larger than your potential energy surface, and so every individual point on this modified potential energy surface is being reweighted, it's reweighted to a different amount according to how much you've raised and flattened at that point. And this is when we go, for example, to the natively unstructured protein stuff, and we calculate our phi-side propensities. In an ordinary MD simulation, you create your phi-side propensities and you put 1 point, you divide up the Ramahan plot into bins, and you put 1 point in each one of these. Whereas in our case, what we're actually doing is that we're doing 1 point multiplied by this weighting factor, which accounts for the amount by which we're raising and flattening the potential energy surface at that point.
Yeah, but how much energy is that per division? If it's both in factor of 200k calories.
Oh yes, it's large. I mean, and there are issues of we have to sample an exceedingly large number of points during our simulation, so we try and reduce the statistical error. And even there are problems that has been reported that one of the difficulties is when you start going to very large acceleration levels, you tend to overestimate to some extent the lower energy regions of the potential energy surface and underestimate the higher energy regions of the potential energy surface.
What happens like when you show, like you have a bit lower, you have a plot on the residual dipole.
Yeah.
That's a couple of slides down there. More. That one.
Right, but all this is already, we've already reweighted.
Yeah, but what about the previous slide? The next slide.
Well, so the reason that you start increasing your Q value is because of statistics. The statistics
is that the reason why? Like all these values that are plotted on the y-axis have been debiased there. But
yes.
But they're all debiased, so you should have the same answer on all of them.
No, because we're sampling a different amount of conformational space.
Let's say you sample of the residue with whatever the value you based it, the sampling is not the issue. The only thing that is the issue is whether your design is correct.
Yeah, I mean, I think this can be addressed by implementing this in a Hamiltonian exchange simulation.
That's been done,
yeah. Okay.
Yeah.
So yeah, I mean, if you ran it out to infinite time, then the unbiasing is perfect. But because you're unbiasing on a limited number of samples, that's why you start getting statistical inefficiency out of hydroxide.
But the reason why it's creeping up above 200 there,
that's statistics.
That's because you.
Well, that's not definitely certain. You can argue that it's statistics, but you can also argue that we're actually sampling far too much conformational space.
No. There's no miracles. There's no miracle. So the idea here is the following. You change the potential and you sample. In principle, like he said, if you have infinite time, everything is fine. But as you change the potential, you want to make sure there are settling the regions that you originally want to settle. If you start to settle now, you deform so much that you're settling the wrong regions, then when you pick it up, you're in trouble again. So that's where you run into trouble.
So do you think that's what Jose is saying, that actually at that point you're distorting the circuit?
I accept the fact that one possible region, and certainly part of the reason for this increase, could be due to this statistical uncertainty. I accept that. But I also think that we have to be careful because residual dipolar couplings are sensitive up to dynamic motions of milliseconds. If we start sampling conformational space beyond that amount of conformational space that is sampled within a millisecond, then we're sampling too much conformational space, and that would mean that the RDCs are going to get worse again.
But along those lines, I mean, is this a universal value then of the alpha parameter or whatever that you're using for all the different simulations, or is it system specific?
Well, the amount of the optimal acceleration parameters are of course system specific, because in some systems you're traversing smaller energy barriers, in other systems you might be traversing a few large energy barriers. But we seem to have found that depending on the size of the system, you can come up with a pretty good guess as to what the optimum acceleration parameter should be. It's dependent on the number of residues. And we've now done this on a series of systems. I haven't shown you all the systems that I've done, but we've got a pretty good idea of what are accurate acceleration statistics that will give us decent acceleration with a minimum amount of statistical error.
Just so what's kind of interesting is that you applied to something that already is pretty much like very much like a rock, like ubiquitin, and something that is very, very flexible, like intrinsically dynamic things. One thinks that each of those kind of has more of a robustness. I'm just wondering about what does this look like on a kinase? Do you get DFG in and out motion? Do you see different combinations of
I haven't done. The person who's done the simulation with kinase is Nikolai Fager. There's a poster over there. He's done a form of accelerated molecular dynamics called replica exchange. It's ideally, replica exchange in simulation is supposed to remove and does remove the difficulties of statistical sampling. But the price that you pay for this is, of course, is that you have to do, AMD is no longer that efficient because you have a large number of replicas and you're throwing out an enormous amount of data because you're not using the data that's in the high-energy replicas. My personal opinion is that obviously if you've got the computational power to burn, to do replica exchange AMD, then definitely do it because you're going to get a better result than doing standard AMD. But I think that certainly when you look at the results that I get from my agreement with RDCs and the like, and the fact that there are other people who have simulated RDCs using various methods, and there are the Christian Wiesinger group with Helmut Guttmüller published a paper in Science earlier this year where they used they used an ensemble average restraint. And they got their conformational ensemble, they reproduced RDCs, but this was now by fitting. And if you actually look at I asked them for their data, because of course I've done stuff with Christian Wiesinger. If you look at the conformational ensemble that I'm getting here, my optimum AMD, and their fitted values, and if you actually compare the Q values, they're almost identical. The difference being that this is done without any restraints at all, whereas theirs is done with restraints. So I am of the opinion that replica exchange AMD is an exceptionally good method, but it depends how accurate you want to be, and it depends how much computational power you're prepared to put into your problem. I didn't have the
protein is too big, it blows up. So everything has its limitations.
Yeah.
You don't mean replica exchange, you mean restrained ensemble, right?
Well, no, I'm talking about two different things. We're talking slightly different things. I'm talking about the poster over there, which is replica exchange AMD, is a great method. But obviously, when proteins get larger and you have to do a large number of replicas and the like, then the computational power significantly increases. And my personal feeling is that doing standard AMD, if one is careful about how one chooses the acceleration parameters, one can get at a very cheap, with very little computational power, one can still get a reasonably good representation of long timescale dynamics in proteins, as demonstrated by my examples here, which has just been standard AMD. Now, as for this ensemble average restraints from Christian Wiesinger, I'm not criticizing the paper. As I said, at the end of the day, they give results that are very similar to mine. So fundamentally, it looks pretty good, certainly for ubiquitin. There are certain issues about the paper that I'm not too keen on. One of them is that in their method, as with all ensemble average restraints, it's very difficult when you're applying pseudo-potentials based on experimental data to extract relative free energies. And in their paper, and this surprised me, bearing in mind it's in Science, there is not one word of relative free energies. They've got their 40 or 60 odd structures, which are their ensemble, and they do not once talk about the relative populations, the relative free energies of these different states, which is obviously going to affect the alignment tensor, which is going to affect the values of the RDCs. Now, in the case of ubiquitin, it might well be the case that because it's such a well-folded protein and that the structural transitions are very small, it might well be the case that actually the relative states probably might have relatively similar free energies and therefore relatively similar populations. I don't know that. But certainly if you look at Lewis Kay's work, it's certainly clear that there are other systems where free energy populations become very important. And when he's fitting to his relaxation dispersion profiles, and he's seeing certain states that are only populated maybe 5% of the time. And that is something that intrinsically isn't in these ensemble restrained methods. It's very difficult to get out relative free energies. Whereas in AMD, we're actually trying to correct for the relative free energies. It might only be approximate, but we are getting an approximate representation of the relative free energies. And we find there's quite a significant variation of several kilocalories per mole between different microstates that we observe. And several kilocalories per mole in terms of population is the difference between 1% and, 80%. Yeah.
Approach modeling an unstructured protein if you don't have any RDC information? How would you know which small regions might be structured?
Oh no, we need some experimental data. I mean, I think that's true. In pretty much all simulations, you need to have some experimental justification that what you're creating is realistic. Certainly if you're doing methods such as AMD, where you're perturbing the system and you don't know how much you should be perturbing it, you know that if you perturb the system too much, you start creating isoenergetic potential energy substances. That means random motion. We haven't really got a key idea about the timescales that we're accessing, so we really need experimental data to tell us something about the timescale. I think it's certainly true that we still, I'm not saying we don't need experimental data, God forbid, but I'm trying to say that the whole idea of this is to try and get an understanding of the sort of conformational space sampling and the mechanisms and the different conformational states, the metastatal states that you might find, and then we can do more detailed analysis possibly to get out free energy barriers and rates and the like.
Can you incorporate phosphorylation into your simulations as you did work with the tower? Could you phosphorylate tower and see what that did to the unstructured population?
Yeah, in principle, this method, the nice thing about this method is it's versatile. You could apply it to essentially anything. But obviously you're not going to overcome the problems of the force fields. I mean, if the underlying force field is not that accurate, this method is not going to make it any better. And one of the things that I'm working on at the moment is, I don't know if this relates to your question or not, but I'm working on a variant of accelerated molecular dynamics in quantum chemistry. So if one wanted to actually look at how a chemical reaction, which would involve a change in the chemical structure of the system, then with the accelerated application of molecular dynamics within a QMM manifold, one could look at that as well. But that's not finished yet.
I had a question concerning the unstructured bits.
Yeah.
When people do residual dipolar coupling, they align their protein with different medium, and then they sort of determine, what they have in mind is that there is a molecular frame, and in the different medium, the frame is oriented differently. But at some point, isn't the concept like falling apart if you have like a totally unstructured peptide? I mean, the idea of getting an intramolecular residual dipolar coupling for a molecule that does not have a structure, what does that mean?
Well, but our general understanding, and I know that there is a lot of controversy about this. I mean, there was a paper published by Shockwell back in Science back in 2004 or so, which was obviously made an awful lot of statements that were possibly not correct. But our understanding is that natively unstructured proteins are more structured than we considered previously. That there are local structural motifs, and that these motifs are actually quite highly maintained, and that therefore it is possible to measure finite residual dipolar couplings, and this is what we are doing within our simulations. But I know there are people who disagree with this. It is a controversial area within NMR.
Right.
I think we should break for the day. I just